Good evening, everyone. Bonsoir et bienvenue à notre soirée de discussion intitulée Innovation Cancer, soins et recherches à l'hôpital d'Ottawa. My name is Ingrid Gingras, and I'm the Director of Integrated Marketing at the Ottawa Hospital Foundation. I would like to begin by acknowledging tonight's event is being held on the traditional and unceded territory of the Algonquin people. Et nous sommes ravis de vous avoir tous avec nous ce soir. We're thrilled to have so many of you here tonight in person for what promises to be a night of invigorating discussion with our physicians and researchers who are changing the future of cancer care for patients. We'd also like to welcome those of you who are watching from home tonight. We're very happy to be webcasting this event live. It's wonderful to have you tuning in. Our three guest speakers will be providing us with the most up-to-date information on cancer care and treatments. They will also share with us the latest developments in research and the role the Ottawa Hospital is playing in revolutionizing care not just for us or for Canada, but around the globe. Les découvertes qu'ils partageront avec vous ce soir ont un impact sur les soins des patients à travers le monde entier. We have a format that I think you will really enjoy. We'll hear from our three speakers, Dr. Auer, Dr. Ong, and Dr. Vanderheiden. They will each take time to share their expertise in their particular field of cancer care and research. We'll then open up the floor to questions. This will be your chance to ask our experts. We expect there to be plenty of questions for these panelists. Please know that if you don't get a chance to ask your questions, you might still have the opportunity once our official program has concluded. For those of you watching from home, you'll be able to ask questions through the webcast by using the chat function. It is now my pleasure to introduce the CEO of the Ottawa Hospital Foundation, Tim Kloot, to say a few words.
Good evening, everyone, and I'm reminded of being at church, and I just want to say there's lots of empty seats here at the front if you would prefer to sit, so please feel free. Thrilled to welcome so many of you here this evening and to follow up Ingrid's opening comments. Each and every one of us share a connection to cancer in one way or another, and we have all been touched here tonight. However, we hope through our presentations tonight to demonstrate how donor support is transforming the face of cancer care and research, which has enabled the Ottawa Hospital to become one of the most impressive care and research facilities in North America. We're at the stage now where the brightest minds want to come to Ottawa. They're inspired to be in a place where care is informed by great research and where we have the best equipment and best facilities for them to practice their trade. We've made tremendous leaps forward in fighting this disease because of the research that's happening here. More and more people are surviving cancer and are thriving after their cancer diagnosis. Care informed by research enables new treatment options and offers patients and their families hope, and that we are ready for them when they need us most with informed care options for their treatment. Philanthropy is an incredible driver that enables the hospital to provide and enable great research and great care. I do want to acknowledge an individual who's with us tonight. He doesn't know I'm going to call him out, that I had the pleasure of meeting about 4 weeks ago. And Gavin Murphy is one of our donors who wanted to make a difference. Where are you, Gavin, so I can talk to you? Okay, I'll find you. There he is. Wanted to make an incredible difference on care and research. And Gavin made an extraordinary $500,000 gift to support radiation oncology and research at the Ottawa Hospital. Thank you. Tonight, you're going to hear from three of our world-class physicians and researchers. They're doing incredible work that is making cancer care in Ottawa the very best it can be, and in fact, we are one of the best centers across the world. We're here tonight because of one family's generosity. The Taggart Parkes family, who were joined this evening, made an incredible $1.5 million gift that will fulfill their wish to continue the advancement of cancer research and to help spread awareness and encourage cancer education. Thank you. I had the pleasure of working with the family and know at the heart of your philanthropy was making our community better. You're recognized in our community for your philanthropy and for your community leadership, and we're grateful that you continue to support the Ottawa Hospital, so thank you very much. There really could be no greater legacy than investments today that inform the future of healthcare delivery in our community. And it's gifts like the Taggart family who are paving the way for the next generation, paving a way for new treatment options for each and every one of us when we turn to the Ottawa Hospital and need care. So we're very grateful for all of you in the room as well who count the Ottawa Hospital as one of your philanthropic priorities, and that you want your hospital to be the very best it can be at that time when you need us most. So we're ready to start our program, and to get things underway, it's my absolute pleasure to invite Dr. Rebecca Auer, Director of Cancer Research at the Ottawa Hospital. Dr. Auer.
Thank you very much for coming tonight to this event. I know many of you in the audience are donors or have been donors. Some of you are my patients, and some of you are both. And I just want to tell you how incredibly grateful I am for your support, both monetary but also just in spirit to keep us moving forward with what we're hoping to achieve. I'm going to just talk a little bit about translating innovative cancer therapies to the clinic, and I am the Director of Cancer Research, and I've held that job for the last year. So right across the street, as you've seen, that's the Center for Innovative Cancer Therapeutics, and that is where the Cancer Therapeutics program, part of the Ottawa Hospital Research Institute, is held. We have 18 scientists, 88 clinician investigators, over 100 trainees there, 400 staff total. Of course, we're part of the Ottawa Hospital, and we are affiliated with the University of Ottawa. For me, it's important because right across the street, which I can walk to in 2 minutes, there's the Cancer Center, there's the Research Lab, and of course the operating room and our patient wards. And so in terms of proximity and sharing ideas and research infrastructure, it's all very close, which is excellent. We do service a very large region at the Ottawa Hospital, and our research institute does as well. And we do have one of the largest oncology programs in the country, and I think the largest surgical oncology program as well. The Cancer Therapeutics program has a number of different major research initiatives that span both preclinical and clinical. And I'm going to talk a little bit about all of these. But they really do interact with one another, and the idea is to translate across that valley between basic science research and clinical research, and ultimately clinical care. So when I came as a cancer director, one of the things that we wanted to develop in the Cancer Therapeutics program was platforms of research. And a platform of research is very different than a research program. A platform of research is really a tool. It's a cooperative, interdisciplinary tool that allows research to happen. So it facilitates that research, and it can be used for many researchers and many clinicians. And it's thanks to unrestricted donor dollars that we can pay for these, because these are not things that people would generally fund through grants, and certainly not things that industry would fund. These are programs that everyone can use to help further all of the research programs. And I'm going to talk about the 4 research platforms that we are in the process of or have developed at the Cancer Therapeutics program. So what if the answer was hidden inside you? Sometimes patients say, you know, can you use my tissue? Can you use my blood? Anything that would help to make your discoveries more understandable or translate to the clinic. So the first platform is called Global Tissue Consent and Collection. Essentially, what happens now is if a researcher is interested in getting human cancer tissue, let's say, for research, they have to go through the process of getting an ethics review protocol, which of course makes sense and we all have to do. Then they have to hire a research coordinator who is then able to consent the patients and then go and collect the tissue and bring it to the lab. The problem is that we have a lot of researchers who are interested in this, and so sometimes patients are going to the operating room and they might be approached 5, 6, sometimes 7 times by different researchers asking for their tissue, and they're confused because they said, I already consented to this, and they don't really understand the difference in the studies. The pathologists are a bit frustrated because there's 5 people standing outside waiting for a piece of tissue. And so, and it's an extra step for everyone. So we basically coordinated this so that the consent can be part of the operative consent. So it is close to being a project that's complete. We now have REB approval, but patients now at the Ottawa Hospital, when they're asked if they're agreeing to surgery, they will also be asked if there's tissue from that surgery, will you agree to have it be given to research, and they can simply check a box. It makes it simple, and it's going to significantly reduce the burden on researchers financially to have that tissue collection, and it's also going to increase the amount of tissue we can collect for research. So we're very excited about this, which should start within the next couple of months. Sometimes I think of research, translational research, as unraveling the miracle. And so, you know, if you think about the very first patient who is treated on immunotherapy or CAR T-cell therapy, for example, a young girl named Emily Whitehead, for her, when she was cured, that was a miracle, the first time it happens. And it happens a few more times, and that's really clever science. And then it happens a few hundred more times, and that's a therapeutic that we would approve. And it happens a few thousand times, and that's now called the standard of care. Everything starts as a miracle for the first patient. And so this is a picture of a tumor cell being engulfed by T cells, which we now understand immunotherapy is one of the most powerful cancer therapies we have. And when we have that therapy and it works, it's not enough to say it works. We have to understand why it works in some patients and why it doesn't work in other patients, so we can then make the therapy even better for the patients that it doesn't work for. So platform 2 really is focused on biotherapeutics, and immunotherapy is part of biotherapeutics. Essentially, biotherapies are therapies that are living therapies, so viruses, like oncolytic viruses that many of you have heard of, and cell-based therapies. T-cell therapies are CAR T-cells. This is the therapy that we have now in the clinic. So in fact, we're the only facility in Canada that has the combined capacity to make clinical grade viruses, so viruses we can give to patients in clinical trials, as well as personalized cell-based therapies. Autologous means it comes from you. So we take cells out of you, we adapt them with viruses to make them better killers, and we give them back to you. We're actually the only place in Canada that can do both of those things right now. So it's really exciting. The CAR T program, so CAR T cells are T cells that are removed from a patient. They're engineered with a virus to attack the cancer, and then they're given back. And they've really revolutionized some of the hematologic malignancies like leukemia. And we've manufactured the first clinical grade of lentivirus to be able to give made in Canada CAR T cells. And we're excited to be able to give them here in Ottawa for the first clinical trial in Canada of that. Infected cell vaccine, I'm going to talk a little bit about that at the end. That's a program that's very close to my heart. But again, it's a cell-based therapy combined with a virus-based therapy. So the idea is we take tumor cells out of patients in the operating room, which is what I do. We infect them with oncolytic viruses that are able to infect tumor cells, but also stimulate the immune system, like viruses are known to do, and get the immune system to attack the cancer. We give those back as a vaccine, a personalized anti-cancer vaccine. We're very excited, and again, we're one of the only places that can do both of those things. And then, of course, we have our oncolytic virus pipeline program. We have some amazing scientists in the cancer therapeutic program, which was started by Dr. John Bell, the oncolytic virus program, where we're looking at new oncolytic viruses that are able to infect, kill, and target cancers. We're also supported by a clinical trials office at the Ottawa Hospital Cancer Centre. It's part of our research program. And we conduct trials for industry, but also conduct trials for the Canadian Cancer Trials Group, and then our own trials from scientists that have developed therapies here in the lab. So we're now participating in 62 Canadian Cancer Trials Group. We enroll 17% of all Canadian trial participants across Canada, so really hitting well above our weight in that. We have 13 investigational new drug trials. These are new drugs that have never been tested in anybody before and are being tested here. We're the top accruing site for 7 of those trials. So again, really the place where new drugs are tested. And 30% of all of those types of trials, the participants are enrolled here in Ottawa. And we've won the IND Team Award 5 times in the last decade. So we really are a place where new drugs can be tested. We have biotherapy, so these cell-based therapy trials. So we've had 133 patients enrolled on oncolytic virus trials. That's the most of any other place in Canada. And 229 patients enrolled in molecular profiling trials. So that's taking the tumor out, doing molecular testing, figuring out what works, and then testing them specifically with targeted agents. 681 patients in immunotherapy trials, and Dr. Ong will talk to you a little bit about that as an immunotherapy expert. And we've enrolled the first patient in Canada in a personalized tumor vaccine trial. Platform 3 is looking at how we can give drugs right around the time of surgery to understand better why they work and why they don't work. So as I said, unraveling the miracle means understanding why a drug works in some patients and not in others. And when we have the tumor removed, it's a perfect time to analyze whether a drug that we think is working in a particular way is in fact working in that way. And so window of opportunity trials are trials where a drug that has a safety record can be given to a patient right before surgery, and then we remove the tumor and we see whether that drug has been working. So this is a platform that's been started by a group of scientists and medical oncologists in the Cancer Therapeutics program, and so we have a few trials in that space to date. Finally, which drug is better? Sometimes patients will have several options, and understanding which one is the best option is sometimes difficult to know. Drug companies are very interested in getting their drug approved, but once approved, it doesn't benefit them very much to compare themselves against another approved drug, because if they're worse, then they wouldn't maybe necessarily be used. It's better to focus on marketing at that point. But it's important. It's important for cancer patients, for example, which drug is the best drug to use when you're nauseated with your chemotherapy? Do we know that? Is there a more cost-effective drug, or is there one that works better? So platform 4 is called the RECT Pragmatic Clinical Trials Program, and this was started by epidemiologist, Dr. Ferguson, as well as a medical oncologist, Dr. Clemens. And essentially it's using standard of care drugs to try and understand which drug is better. And these are embedded in routine clinical care, and a lot of the data from these trials is derived from our medical health record, which, as you may have heard, we've got a new health information system at the Ottawa Hospital. So these are real-world trials that we can get the information right from the medical record. And this is actually an old slide. We've now enrolled over 2,000 patients since just starting the program in 2014, which is really much more than any other clinical trial program across the country. And we're also leaders in digital health technology, and with our implementation of Epic continues to be so. And this is, again, a platform that we can use to gather information to understand which drug is better, and to look at new innovations in care and understand how they can improve care. Lastly, I'm just going to talk a little bit about the infected cell vaccine platform, which is very close to my heart. It's part of what I study in the lab. And peritoneal carcinomatosis is a disease where tumors spread throughout the abdominal cavity. And it's unfortunately a common terminal feature of many abdominal malignancies, including ovarian cancer, also colorectal cancer, stomach cancer, pancreatic cancer. And you can see from that picture, this is tumor that's small little drop tumorlets that are spread throughout the abdominal cavity. They create a lot of fluid, not able to treat them with surgery, and they don't respond well to chemotherapy. As I mentioned, our approach is to remove some of that tumor, infect that tumor with a virus that stimulates the immune system, and then give it back as a vaccine. And this stimulates our T cells, which we know attack virally infected tumor cells, as well as other tumor cells that are not virally infected because they are now primed to attack. And natural killer cells, which are just really good killers when you stimulate them with virus. And this is something called a survival curve. It's not in humans, but I hope someday we see something like this in humans. But essentially, we give the animals, in this case, it's a colon cancer, and then we treat them with the infected cell vaccine. So we start treating them on day 10. And you can see from the curve, the black line, every time there's a dot, that's someone that has, or a mouse that has died in this particular case. And you can see they start dying around day 15 to 20. The ones that are alive, we continue to treat. And the orange bar is those mice that were treated with the infected cell vaccine. The black bar is mice that were just treated with tumor cells that were irradiated to stimulate basically a non-effective vaccine. And you can see all the mice that were treated with the infected cell vaccine are still alive. This is just a picture, an MRI picture. Do I have a pointer? No, I don't. So there's actually I can't, without a pointer, I can't tell you. You'll have to trust me. I'm not tall enough. You can see there's a tumor there. It's bigger than the kidney, so that's a pretty big tumor for a human. And that's a week after treatment. It's actually a little bit bigger, and it's got a lot of fluid around it because it's inflamed. But what happens over the next 6 to 13 weeks is that tumor completely goes away. Even though the treatment stopped by about 2 weeks, we haven't given any further treatments. The immune system is attacking that tumor, and actually by the time week 13 comes around, there's no tumor left. So we're very excited by this. It's not in humans yet, but fortunately we do have the capacity in Ottawa to do this clinical trial in humans, and that's of course our next step. So I'll just finish by saying the Center for Innovative Cancer Therapeutics, it's really focused on the translation, and in particular translation of biotherapeutics. We're doing research that makes tomorrow's healthcare possible today. And just a little picture of a surgeon with a heart, but just research is really part of care. And healthcare has care in it for a reason. And I think one of the things I'm struck by when we've implemented this new health information system is how everyone, all the doctors I work with and the nurses, they still put care well before everything else, because I think that ultimately that's what our job here is. And research does provide that care. It provides hope for patients. It provides opportunity to understand the disease, which is important to patients, and provides care. So thank you very much. It's now my pleasure to introduce Dr. Michael Ong, who is an expert in immunotherapy and a medical oncologist here at the Ottawa Hospital.
Thanks, Rebecca, and thank you all for coming out today. It's a pleasure to be introduced by Rebecca, who she and I almost 20 years ago, I was a medical student on her general surgery service as she was a senior resident. And look at us now, still working at the cancer problem. So today I'm going to talk about something that could be just as exciting as the Raptors playing in the NBA Finals, Game 6. Which is cancer immunotherapy. I'm a medical oncologist, so I treat patients with what we call systemic therapies, really drugs that we think that can be helpful in cancer treatment. My particular specialties are skin cancers and urological cancers, and I'm one of the drug development specialists as well. So I do first-in-human trials of new promising drugs that Rebecca and her team might develop. So as medical oncologists, we're part of this facility here at the Cancer Center, but we deliver care to the wider Ottawa, Champlain-Lynn And we basically based out here at the Cancer Center, but also at the Irvin Greenberg Family Cancer Center. But we also order all of our treatment out to anywhere, Pembroke, Renfrew, Winchester, Cornwall, etc. So what's nice about this region is we can centralize care. We really have a handle on what's happening within the region. And immunotherapy, even though it's a very new entity, we are now, in the last year, really delivering all of this at all of these sites. And I think that's quite an accomplishment. So I think, three questions here is, main question is why use the immune system for your cancer? And I have three simple answers for you. First of all is the immune system knows you best. Okay, one of the challenges that we have with cancer treatment is trying to spare people side effects. And to do that, you have to have a treatment that really can differentiate the person from the cancer. So the immune system is quite good at that, very good at that, in fact. Immune system can also adapt as cancers adapt, which is a very important aspect because cancers by their nature are unstable. They tend to change with time. So that's something that's important. And the final thing is that your immune system can also remember. So what is unprecedented with some of these immunotherapies is that the effect of the immunotherapy can last even after we stop treatment, which is a really key feature because if you're a patient with cancer, I think one of the last things you want to be on is ongoing treatment unless absolutely necessary. So what we found in recent years is that actually there is a natural immunity that patients have to their cancer. It doesn't always happen with every patient, but it can happen. And one of the key mechanisms that cancers actually try to protect themselves is they cloak themselves in something called PDL1, programmed death ligand 1, which essentially hides from these T cells that Rebecca was talking about. And that's one mechanism that cancers have been, even though you had a natural immunity, hiding from that. Now we've been able to give these new medications called anti-PD-1 drugs, and there are many of them. And suddenly the immune system can do what it wants. It can attack this cancer that it had actually recognized already. Now, this discovery, as you may know, won the Nobel Prize back in October just last year. And there's a more complex diagram here, but essentially, you need one mechanism to train your immune system, another one to prevent this kind of cloaking that I talked about. And both of these are right now what we use in the clinic. Why does this work? Well, cancers by their nature, they can actually mutate and change. And it seems as though if you have many, many mutations, the more mutations you have, the more likely it is that your body can actually see this cancer as foreign and attack it. So in the red bar at the top, those are cancers with a very high level of mutation. And in the green, it's those with a lower level of mutation. And there seems to be an association between higher mutation burden and response to some of these treatments. What's also interesting is that these higher mutations can span many different types of cancer. So even though the cancers that I treat, like skin cancer and kidney cancer, skin cancer and say bladder cancer have a very high mutation load, there are some patients, for example, with colorectal cancer, that because of their biology, they also have high mutations and they can benefit from immunotherapy. So here are some examples of responses in my patients. For example, here's a patient with melanoma, metastatic, multiple spots in their lung, as you can see by these red arrows. And then after a combination of 2 different immunotherapy treatments, 4 months later, it's almost all gone. Here's another example of a patient of mine with a rarer cancer called Merkel cell carcinoma. This happens in patients with a lot of ultraviolet light damage and immunosuppression. And this is after just 2 months of treatment. It was a more locally advanced lesion, and we treated it with immunotherapy. And by the time the surgeon got to this patient to consider surgery, they weren't quite sure whether they should do the surgery at all. So that was an interesting scenario to be in, where Merkel cell carcinoma, the data for this kind of immunotherapy is just coming out. So we're trying to understand what to do in these situations. So this is a graph of how immunotherapy is now being used at the Ottawa Hospital. This is data from our own cancer center, and you can see that this is a very new treatment. We've only really had this uprise in the immunotherapy use since 2015. I would say that in large part, the early access to immunotherapy for patients has been through our clinical trials office. And as Rebecca has pointed out, over almost 700 patients put on those immunotherapy trials. And now many of those have become standard treatments. This is not showing up well at all, but essentially, I think the ways that we're leading in the immunotherapy is we're the leaders of a trial asking the question, how long should we be giving these immunotherapy drugs? Should we stop at best response, or should we continue on and on? And that's a Canadian-wide study, and one of our clinicians here is the national lead for that study. Very important study internationally. We're also looking at studies of how to minimize and prevent side effects, how to integrate immunotherapies with radiation or other existing treatments, like, for example, oncolytic viruses. We're looking at combining surgery approaches with immunotherapy before or after to boost cure rates. And we're also looking at immunotherapy in orphan diseases that may not have access to it as well. So I think our clinical trials office and all of our operations here, I'm just one member of a large team here, are really looking at all these areas. And so I'd like to thank you all for coming today and for your support of our organization. So next, I'd like to introduce Barbara van de Heijden to the stage.
Thank you, Dr. Ong, and welcome, and thank you for coming. I'm the researcher. I'm not a clinician. I will not be showing you pictures of real cancers, which is good for me, and especially if you've had something to munch on, it might be easier on your tummies. So one of my jobs is being the Corin Boyer Chair in Ovarian Cancer Research. That's a position that was established 20 years ago, almost to the day, and I accepted the job in 2000. So I've been doing this for 19 years. When I started, I was very lonely because there were exactly 2 other ovarian cancer scientists in the country, one in Vancouver and one in Montreal. And so we put our heads together and we decided we can't do this just 3 people. We will never make a difference. And so we've spent the last 20 years building research capacity for ovarian cancer across the country. And I'm pleased to say that we now know a lot more about ovarian cancer than we did 20 years ago. And last year, finally, we had the first new treatment for ovarian cancer be approved in Canada in more than 20 years. That's just the beginning. Now that we know what we're doing, we plan to have a lot more coming out in the next few years. So Dr. Auer has already pointed out that there is sort of the cogs that she had, and they're all interconnected. My lab tends to live around this one area called basic research, but when we discover something that we think could be a good cancer treatment, we need to test it. And so my lab for the past 20 years has been developing model systems for preclinical testing of really promising new cancer therapies. And as they start getting better, we needed to move things into that pipeline that is translational research, taking it out of the animal, the mouse models usually, and putting it into human tissue. For that, we had a tumor bank that I established in September 1995, and I am over the moon that Dr. Auer is establishing this global consent and collection process so that my technician will not be running back and forth to the OR every time we get a page. We now have a much more efficient system coming online, and we can focus on the research and not the exercise going back and forth to the OR. But it has allowed us over the past 20 years, or 24 years, to use human tissues. When a woman with ovarian cancer has her surgery, those samples can come to us and we can test our new treatments directly on the human cancer cells. And anything that is most promising then gets shunted over to the likes of Dr. Ong and Dr. Auer into the clinical research venue and ultimately into clinical trials where we can really see whether this is going to work. Now, all of these pieces together require a lot of expertise, a lot of different people with different expertise. And I've been really fortunate in the cancer program that while I came on board in 2000, I've had the great pleasure of working with a lot of different individuals who have different areas of expertise. The first row across the top are the 5 gynecologic oncologists. So if you've had any type of gynecological cancer, including ovarian cancer, you will have been treated by at least one of these individuals. They're the surgeons. Then the row next to them, below them, are the pathologists. And these have been incredible collaborators and still are for much of our research because they help us look at a human ovarian cancer and tell us what we're looking at, because that's not my area of expertise. What is my area of expertise is along the bottom with all those other scientists. And it used to be just Ben Sang and I. And over the past few years, we've been pushing, prodding, encouraging, and basically trying to stimulate interest in ovarian cancer in many of my colleagues. And there are now a lot of us in the Cancer Center that are working on various aspects of ovarian cancer novel treatments. So it's been a very exciting time as we've grown, even locally, but across the country as well. As I said, we've built this capacity across the country. In 2002, I organized the first Canadian Conference on Ovarian Cancer Research here in Ottawa. And it was a small, mighty group, and what we decided is we needed more. We needed more expertise, we needed more people working on the problems that we had to deal with. And so from that point on, every 2 years, the conference has done the politically correct thing in Canada and bounced around to different cities every other year. And I'm very excited by the fact that for the 10th Canadian Conference on Ovarian Cancer Research, it will be returning to Ottawa, and I will be hosting that meeting next June. So I'm looking very much forward to being able to look back over the past 20 years and see how much progress we have made and make plans for what we're going to do for the next 20 years. So when I first started, we were asking the question, where do ovarian cancers come from? And that might sound like a really silly question because they come from the ovary, of course. But about a decade ago, we found that that's actually not true. Many cancers do come from the ovary, but many ovarian cancers actually start in a structure that's right next door called the fallopian tube. Who knew? And how we deal with it and how we study it and how we understand how it develops is completely dependent on where we're looking. So now we have to look at both the ovary and the fallopian tube. And we've been doing that and trying to find ways to understand how they become cancer. That gives us some tools to identify methods that we can maybe prevent this, and if not prevent it, at least be able to detect it early. The rest of my lab all focuses on the testing of novel treatments, and I'm going to tell you a little bit more about one of the stories that we have. I only have time for one story, but it's a good one. And so we've been studying a process called epithelial mesenchymal transition. Big mouthful, we'll call it EMT. And what you see on the left side here are these nice square cells. Those are epithelial cells. They very much attach to each other. They like to stick together. And when they stick together, they will grow as a tumor, but they don't move. They basically just grow in place. But the thing is that cancer cells have a tricky ability to transition into these much flatter, elongated cells that you see on the right. And those cells don't talk to each other as much, and they migrate. They can move. And what they do is they move out away from the primary tumor, and they can move to other places. They can move into the blood system and just into your blood vessels, migrate to someplace else in your body, come back out of the blood vessels and cause a secondary or metastatic tumor. So mesenchymal, or these migratory cells, are responsible for many of the metastasis that happens. And we thought this might have happened in ovarian cancer as well. We had a hard time studying it because ovarian cancer is quite complex. But then, without a donation to the Ottawa Hospital Foundation, that's where the story would have ended right there. but we have more to tell now because we have received funding that has allowed us to investigate this much further. So this little funky diagram is one kind of epithelial ovarian cancer cell. In its epithelial state, it's in blue, and each little dot is one ovarian cancer cell. So there's a whole bunch of them there. They're all in blue. We found a way that we can induce these epithelial cancer cells to become more mesenchymal, to make that transition, that EMT. And what you see is on day 1, they're in the orange space. On day 3, they're in the green space. And by day 7, they are fully mesenchymal and they're in the red space. So they go through this transition to go from blue to red, from epithelial to mesenchymal. So that's cool because every single cell that you see up there, what's showing is all of the genes that are activated in each of those cells. So we can learn a lot about that process. And what we learned was when we started looking at what genes are activated as cells go from epithelial to mesenchymal, is there's a lot of genes all on the right-hand side. I won't describe them all, so you'll be pleased with that. But what happens is that as it goes from black to purple to brighter purple or pink, that is genes being activated. And you can see all of these genes being activated as they go from left to right, as they go from epithelial to mesenchymal. And the important thing about all those genes that you see on the slide, they're all regulators of the immune system. This stunned us because these are cancer cells producing molecules that control what the immune cells do. So this was really stunning. And was like, okay, what do we do with this? This is really fascinating. And what are the consequences if these cells were actually a tumor and they're producing regulators of the immune system? That's maybe how they control how tumors will progress. And so we have been the first in Ottawa to use this new technology called single-cell RNA sequencing. And we're using this technology to build what we're calling an atlas of a collection of human ovarian cancers. And what we're doing is we take a sample of a tumor that a patient is giving up during their surgery. We bring it to the lab and we take all the cells apart. We dissociate it. So now we have a collection of single cells that are all the cells that make up that tumor. Then we do what's called sequencing all the activated genes that are present in each and every cell. And then we can make a plot that looks like what you see on the right. Now, you see the big pink blob in the middle? Those are T cells. Those are the cells that both Dr. Auer and Dr. Ong have talked about. Those are the cells that are supposed to be fighting the tumor. What the heck are there so many T cells doing in a tumor and it being a tumor? Why are they not killing the cancer cells? And this is why we can take these cancer cells, which are the blue on the bottom, and all these other types of immune cells, which are almost all the other colors that you see, and try to figure out what are the cancer cells doing to control which immune cells are present in that tumor. And what is it doing to stop them from acting, right? Dr. Ong mentioned PD-L1. It's a cloaking mechanism. It's quite clear that PD-L1 is very likely to be in that ovarian cancer. But maybe it's not alone. Maybe there are a number of other inhibitors of the immune system that is produced by the ovarian cancer cells. And that's the reason why most immune therapies that have been tested so far, well, all immune therapies that have been tested so far rarely work in women with ovarian cancer. 10 to 15% have some form of response. Most do not. So we don't have the right immune therapies yet. But this kind of information, where we know how cancer cells are controlling the immune system, will allow us to find out and control what are the cancer cells doing? What are all those factors they're producing? And can we block those factors that suppress the immune system in order to make a much better, more effective treatment for ovarian cancer? That is our goal, and we hope to have it achieved in the next few years. And I thank you again for anyone who has contributed to this research. I hope you are pleased with the outcomes and the directions that we're taking going forward. I do need to say that I am not alone in the work that I do. The diagram on the right is the entire research team at a recent lab barbecue. We have a big barbecue every summer. There's a lot of brilliant minds in that group and a lot of out-of-the-box thinkers, and that allows us to do things that I normally wouldn't dare to do alone. And you will also see on the left-hand side the group where we're wearing our teal ties and tutus. We walk with patients every year in September for the Ovarian Cancer Canada Walk of Hope. And if you want to learn more about our research, just come to the walk and we'll tell you all about what we do. Thank you.
Okay, thank you so much, Dr. van der Heijden, Dr. Ong, and Dr. Auer. I'd actually invite the two of you to come join Dr. van der Heijden here on the stage. We're going to get started now with our Ask the Experts part of tonight's program. I have two foundation team members, Hannah and Rhonda, who are here tonight, and they will help guide the audience. They will have the microphones. Don't steal it away from them and start, your solo singing. They will help me guide the audience. If you're watching through the webcast, now's the time where you can also start putting in your questions, and we will moderate from here. So I will ask, I know it's going to be hard, but I will ask that our panelists keep their answers concise so that we can get as many Q&A questions and answer possible. And again, if at the end of the night you feel that you just have this question that you absolutely have to have answered, we'll be around, and if our panelists have to leave, then we'll be sure to share them with them and follow up with you. So I will look to the audience to see if anybody is going to be the first. Oh, thank you. We don't have it. Thank you. So Hannah, do you want to come over here, please? You don't win a prize for being first, but I certainly appreciate it because I didn't have a song prepared.
Thanks. So somebody has to be first. So
someone has to be first.
I'd briefly like to ask you about early detection, as I noticed it in one of the slides there. What would you consider to be some of the main challenges to early detection of cancer in your fields, and also some of the achievements or successes in early detection? I guess we all have experienced, those who have been touched by cancer, issues around, could we have discovered it earlier? What obstacles have been in the way of discovering it earlier? And that's an issue that I'm just interested in your reflections on, your brief reflections. Thank you.
So challenge of early detection and then a success. Who would like to go first?
If my microphone works,
there we go.
I can take a stab at it. Absolutely. I mean, I think we want to be able to detect cancers at a stage where we can actually make a big difference. And I think that examples of early detection strategies could be, say, in people that have had a heavy smoking history of a certain age, doing CT scans. I think some of the challenges you come up with is the number of people you need to subject to that particular test in order to, say, cure X number of people earlier. And that's a lot of extra people that had to be scanned, for example, to actually try to make a difference. And you end up, say, with a handful of people, like say 5 out of 100, that might have been benefited by that strategy, and yet you have another 95 patients that had CT scans every 6 months or every year. And so those are some of the challenges that we face. So it depends on what strategy we're using. There are some very promising technologies, for example, like doing a blood test, for example, that you can now, with very high sensitivity, find that there might be changes in those blood tests that might lead to another test that might confirm something. So I think that the cost of some of those new technologies is ever going down, and our ability to detect things is improving. And I think in the future, we may be able to integrate that into testing, but I think there are also many, many different types of cancer, so one strategy can't fit all of those, and not yet, at least.
Dr. van der Heijden, do you want to follow up with that?
Yeah, so I think that's the key, is that no two cancers are alike, really. So it's hard to find something that's going to be able to be applied to all cancers. I know in the ovarian cancer field, we can identify women at risk because those who carry BRCA1 mutations or BRCA2 mutations, these are mutations in your genes that put you at a higher risk of developing breast or ovarian cancer. So once a person has been identified or has a family history where they can have that mutation identified, then they know that they are at higher risk. And they will be followed much more closely. It's regular ultrasounds. There is a blood test, but it's not particularly reliable, but it's used for women at high risk. So it's just very, very close monitoring to see if they're able to catch it before it begins or as soon as it begins. And more importantly, the prevention I put up there is the reason we now use prevention is because fallopian tubes can easily be removed after a woman has had her children. She doesn't need her fallopian tubes anymore. She doesn't even need her ovaries for that matter. She can take them out, and that reduces the risk very substantially. So those are the methods that are being used in women that we can identify who have a high risk of breast or ovarian cancer.
Dr. Auer, do you want to touch on any successes that you'd like to share from that early screening and prevention?
Yeah, so I mean, I think that for any early screening test, you have to have two things. You have to have a sensitive test, so you capture anyone at risk, and you have to have a specific test. It has to be specific for cancer. And the problem right now is we have some tests that are very sensitive, but as we said, then a lot of people will get a positive test and have to undergo further testing, and it's not very specific, or vice versa. I think colorectal is one area that we do have a fairly good test, a FIT test, which is being rolled out in Ontario now, and it's checking for blood in your stool. And that will lead to a colonoscopy, and it is quite a good sensitivity and specificity. But I do think that both Dr. Ong and Dr. Vanderheiden touched on blood tests. There is almost certainly in the next decade going to be some emerging blood tests that will allow us to have a more sensitive and specific way to triage people's risks. And they may involve looking at changes in the immune system or looking at mutations in the DNA that floats around in your blood. And these are things that are being studied in research protocols now. And so I think the future for early detection is actually quite bright.
Okay. Now the hands are going up. See, we knew it was bound to happen. Rhonda has a question in the back. Yes. We'll swap from one side to the other. No fighting, okay? There's a line. You picked your side when you sat down.
My question is about immunotherapy. We've all heard it is going to be the next best and brightest thing, and certainly I've seen it applied personally to a friend with lung cancer and another one whose cancer began as melanoma. But being completely selfish as a breast cancer survivor, I'm just wondering, I've never heard immunotherapy as it relates to any breast cancer treatment, so I'm guessing that your earlier answer of no two cancers are alike is probably the answer, but I'm just wondering if there's, in the future, we're going to see immunotherapy applied to breast cancer or many prostate, whatever other cancers as well, and see the kind of success that we've had so far with other cancers.
Dr. Ong?
So globally, this is an absolute hotbed of research, right? I mean, we know now, just in the last 5 years, that we can have great success in some of the tumors you've talked about, and not other major tumors like breast cancer and colorectal cancer. And the question is, why not yet? And in breast cancer, not even all breast cancer is the same, right? You have hormone-sensitive breast cancer, HER2 positive. And now in triple-negative breast cancer, there is this sense that immunotherapy may help for some of patients with that kind of scenario. So I wouldn't say not all breast cancers. But I think that is that's the next frontier, and there's been thousands of clinical trials that are open or been conducted in this area. Should we be combining another therapy of some sort, whether it be radiation or targeted therapy or chemotherapy or something of the like, and will that complement immunotherapy? Also, I think we have an incomplete biological understanding, and that's where the scientists really matter here. It's like, what are we not understanding about why immunotherapy doesn't work yet? So there might be another target. So I think that is very critical to understand the biology and link that to the clinic, and that's what we hope to accomplish here.
Okay, a question online for Dr. Auer specifically. What is the biggest barrier to pursuing research? Examples given: money, time, people, power, right? Not to put you on the spot, but you're sitting in the seat, so we're going to do that.
Yeah, I mean, there are a number of barriers, obviously, to being able to, drive research. I think conducting research is certainly I think we're doing a very good job, a collaborative job of getting research done, but you always want it to be faster. You always want it to be moving that much quicker from an amazing preclinical discovery to a clinical trial or to a standard of care in a patient. And I think that's where the frustration lies. And I do think the barriers are, money is certainly a barrier. These are very expensive, which is why often companies get formed, and then companies get involved, and then companies buy other companies, and companies have different ideas of what they want. They are obviously they're interested in curing patients, but they're also interested in making money. So that becomes a factor. And I do think the regulatory agencies also provide barriers, and those are in place to protect patients. I do sometimes, we do sometimes struggle with that because when you're protecting a patient who has just a few months to live, we have to be very careful about weighing the benefits and the risks. And I think when HIV, for example, there was many barriers in HIV studies, the HIV community just sort of stood up and said, no more, you have to let us move forward. And that really did change the regulatory landscape for HIV and has made HIV a chronic disease instead of a death sentence. So I think that's also happening in cancer, and patients are saying, you can't put up barriers to treatment. We need to be able to have access to these, and I think those are important as well.
Okay. Somebody from this side? Or gentleman right behind you. Go ahead, Hannah.
Dr. Van der Eyden, I was interested when you were talking about the low percentage of ovarian cells that reacted positively to immunotherapy. And I have a vested interest in another cancer, which is awkward. Grandfather, father, sister, and cousin, all of whom died of pancreatic cancer, which I understand is also resistant to the same type of trials. So I'm asking, I know that your interest is ovarian cancer, but do the research centers across the country share these techniques? And please God, is there a pancreatic cancer researcher who's interested?
Oh, there absolutely is. And I don't have to go across the country. I can just go down the hall and talk to Carolina Ilkow. So, and we meet on a weekly basis. We have 2 meetings a week, one of which we share the research that's going on. We take turns talking about the research we've done, and everyone in the room is thinking, okay, in my project, how does that fit? And so when I'm talking ovarian, Carolina is listening for her pancreatic studies. When she's talking pancreatic, I'm listening for relevance to my ovarian studies. And we've got a room full of 50, 60 people who are all listening and trying to exchange that information and see how it's going to fit with the studies that they're doing. So there's internally a lot of information exchange. And of course, we have these conferences. Actually, the next National Cancer Conference is here in Ottawa in November. And so all the cancer researchers of all different kinds of fields, of different types of cancer, will be coming to Ottawa. And so that would be a massive 3-day exchange of information. So a cancer researcher, a researcher period, cannot work in isolation anymore. It's just not possible. You can't move fast enough, forward enough. So, no worries.
Dr. Auer, can you explain a little bit about the uniqueness that we actually have here at the Ottawa Hospital with clinicians and researchers working so closely, because it's not common across the country?
Yeah, I mean, one of the nice things about Ottawa is that we do have proximity, and we also have centralization. So, obviously Toronto has a pretty big reputation for research, and they are much larger than we are, but they're also fractured. So one of the nice things here is all of our cancer patients, for example, do get centralized here. Now, of course, they may get their treatments elsewhere, but it's under our supervision. And so that actually allows us to be able to enroll better in clinical trials, which is why I think one of the reasons why we hit well above our weights in terms of clinical trials, because we do have that centralization. I also think that there's a culture in Ottawa, and I went to school in Toronto, so I know a bit about the culture there, that is very collaborative. And I would say that we know each other on a first name basis in the Cancer Center. Our kids play soccer together. Our families go to the same schools. And so we have a relationship that it goes well beyond the science and well beyond the patient. And I really do believe human relationships make things happen in a much better way than, let's just say, data and dollars do alone. And so I think we do have this proximity to each other and this collaboration that allows us to really put the patient first. And it doesn't really matter who gets the credit as long as we're moving the science forward. So
That's great. Rhonda has a question in the back.
Well, I'm very pleased that there is a research team for ovarian cancer. It's like you never hear much about what's going on with ovarian cancer. It seems the other cancers seem to grab the limelight. So I want to say thank you. And I'm curious why it took so long, or it seems to have taken so long to get research going on ovarian cancer. Probably there's no right answer. But thank you for your work, and I look forward to the conference next year to see what's happening.
We actually do, for the conference next year, we have 2 affiliated workshops that we organize at the same time. One is for all trainees who want to come into the field, so we can give them a crash course on what is ovarian cancer. They actually meet with patients, so they hear firsthand what those experiences are. But the other full-day workshop is all for patient survivors, advocates, their families, to learn about the latest in research in ovarian cancer and sort of what's coming very soon to a clinic near you. So if you get in contact with me in any way, I can make sure that you are informed about this workshop when it takes place next June.
I think we can also share information about that conference with everybody tonight who's RSVP'd when the time is right and you're all set, ready to go. So one of the 3 of you here, what improvements in cancer care do you foresee in the next year or the next 5 years, or essentially in the next little while, because I know a year is very quick. We're almost July, in case you didn't know, so the time is passing us by. Anyone want to start here?
Well, I mean, it's happening worldwide at an almost staggering pace. So as a clinician, I feel even treating 2 different tumor types, I'm behind in the data, right? In Chicago, a world conference already happened, and there's thousands and thousands of research studies that poured out, and you basically have to go through and look through all that because you have patients that are in that scenario, and what has changed. And so there's a lot of data that some of the big data that we can make, will make Health Canada changes and will be an approved medication in Ontario, and I foresee for sure that some of those changes will be made. And I'll give you examples like whether or not we should combine chemotherapy together with immunotherapy as first treatment of bladder cancer that's recurrent, for example. And now we're currently conducting studies of combining those treatments even before surgery. We've opened up a study here to see if we can, nip it in the bud right up front, rather than waiting for it to recur. So these are like changes in practice are done through clinical trials, and we are conducting a lot of those, many of those.
So good things will happen shortly.
Well, through a lot of effort, not just at the Ottawa Hospital, it is a global affair right now, to try and find out what works, and that only happens through meticulously documenting what happens when we do this treatment in this scenario and what are the outcomes. And so that's an answer to a question of why does progress seem like it's slow sometimes? It's because we have to go through the rigor to prove that it works, and that is a very expensive and time-consuming affair. But progress is being made, and I don't know how old I look or young I look to you guys, but in the time that I've been an oncologist, there's been a ton of progress that has surprised me, and there's much more to be made, and we know we're not there, but we're stepping in the right direction. So I think it is very important.
Oh, Hannah has someone. Okay, go ahead.
I was always, we were sort of strong supporters of Dr. Bell's viral research, and I'm just wondering how the immunotherapy complements that, or how it works. I would like to know that.
Sorry, I can't find this. Oh, there.
So maybe I can address that. So I think it's an excellent question. And I think when Dr. Bell, so I was in the lab actually as a resident, and I did my master's with Dr. Bell when the very first sort of major breakthrough came out with oncolytic viruses in his lab. And he really is, as you know, one of the world leaders in that field. And at that time, it was all about the virus being able to find the cancer cell and uniquely infect the cancer cell and attack and kill the cancer cell. And the reason why was because cancer cells are fundamentally different than normal cells. And so all of the lab was really focused on trying to suppress the immune system from attacking the virus so that we could infect the cancer cell even better and kill those cancer cells. And then I finished my residency and I went off to New York, and I came back and everything had changed in the lab. All of a sudden there had been this realization that in fact that's only a really small part of the story. And what's even more important is that it's not so much that the virus can seek out every single cancer cell and kill it. It's that the virus can attack and infect the cancer cell, and in doing so makes the immune system suddenly realize that that cancer is foreign. So just as Dr. Ong said, cancers do have some innate abilities to pretend that they're just like the rest of your cells in the body. Because they are formed from your own cells, they kind of hide behind a cloak. PD-L1 is one of those cloaks, but we know there are many other cloaks that they hide behind. But nothing stimulates the immune system better than a virus. And so if you infect that cancer cell and present all of those foreign things to the immune system wakes up and takes notice. And then if you can combine that with sort of traditional immunotherapy that we now have approved in the clinic, it's sort of like the oncolytic virus turns the car on, and the immunotherapy or the anti-PD-L1, anti-CTLA4 takes off the brakes and puts on the gas.
Oh, just a second. Did you want to pass the microphone again?
So are they working together then, the immunotherapy and the viral therapy? Are they working hand in hand? Is that what you're saying?
So I think having been an investigator on some of these trials, actually an investigator on the single agent oncolytic virus, I know that we had learned a lot from doing those initial studies. We did a first in human study with that. And I know that what's then gone on is now there are trials of involving virus with some of these immunotherapies, and that's still ongoing and data being generated. So it takes time. These are pan-Canadian studies. There's a lot of regulation, and Ottawa's not unique in trying to test viruses. Like, for example, for melanoma treatment, there's the first FDA-approved oncolytic virus, TVEC, which is used as an injectable into tumors, has been proven to improve survival. And we've been conducting trials with the TVEC with the immune checkpoint inhibitors, and yet we're still waiting for those results of a trial we had conducted 3 years ago. So it takes time for these things to happen, but these are like investments that you make, and then we're hoping that the results will be favorable. So that's how it goes.
Okay,
Rhonda?
thanks. Exciting.
Hi, good evening. In all of your presentations tonight, you've talked about clinical trials. I was just wondering, how are patients picked for these trials? Because you've all talked about different trials and clinical trials. I'm just wondering how the patients are picked for your trials. We have a clinical trials team, right, that we have to actually pick trials for the cancer center, right? We actually try to have a trial that would fit nicely, like we need a trial in this scenario because we need to have improvement in this particular area, right? And we also pick an area that we think we have appropriate patients for, right? Then anybody that is in that scenario and we think they're appropriate, we will present them, if possible. We're interested in accruing patients, but there might be barriers, right? There might be that the patient's not fit enough because of other medical issues, or their lab tests are not good enough, or maybe they live too far away. We don't automatically exclude them, but if they're living in Pembroke, they may not want to come every other week to the cancer center here. And the trials have to be done in a regulated center like this. So we try to accrue to the trials whenever we can, for the right patient.
Did that answer your question? Yes. Okay. Rhonda, go ahead.
Just a very simple question. You're represented here by the Cancer Foundation, the Ottawa Hospital Foundation, I mean. How much is money, just the plain, simple availability of money, affect your ability to be able to move forward?
Dr. Vander Heijden, do you want to take this one?
So there's no way we would have done this study if we hadn't received funding. I can tell you that second last slide I showed, where we were doing single cell sequencing to be able to look at how cancer cells and all the different immune cells are present in a tumor. One tumor costs us $5,000 to do that analysis. It just, right from getting the tumor to taking it apart, the sequencing, and then the analysis afterwards, just the simplest form of getting that tumor and analyzing it is $5,000 per patient. So it's not a trivial thing. We've applied for money and we're waiting to see. My goal in the short term for the 1 year was to have this atlas done in a year. We started this 3 months ago. We have 3 samples done and we want to do 12. So if we get enough funding to get the rest of it done, we will finish it within the year. So it's an expensive endeavor. The student is funded by a scholarship, so I don't have to pay him. He's brilliant, so he's just eager to move on this. There's no shortage of talent, I have to say. The postdocs and the graduate students and even the undergrads that we have here in Ottawa, they're brilliant. They are really smart, but we have to pay them. We don't pay them much. They get barely above minimum wage, but they have to be paid nonetheless. And that's probably one of the limiting factors for me is paying the staff I need to do the experiments.
Yeah, I think my answer would be it depends on what you're trying to accomplish and do. We try to use money wisely. But for example, to give you an example of costs incurred, if I've set up a pan-Ontario database for all prostate cancer patients in all of Ontario, and setting up that database in the ICES network costs, that's about $25,000 to have that kind of database and about 6 months of work. I've also tried to set up a clinical trial of a pilot study of only 25 patients, combining radiation with immunotherapy before bladder cancer surgery. That costs minimum $500,000 for just that pilot 25 patient study. So it really depends on the context of what you're trying to do. And research costs can escalate just dramatically, especially in the clinical world. And that clinical trial is talking about all drug costs paid for, by a sponsoring pharmaceutical company, but it's all regulatory costs. So it's the cost of somebody monitoring the study and making sure at 3 sites it is being conducted properly. And that's some of the regulation that Dr. Auer is talking about. It's very costly.
And as another example, there's the cost of the basic science, there's the cost of the trials, and then there's the cost of making, for example, in our biotherapy manufacturing suite, even just to make a batch of virus to be used for a clinical trial. That doesn't include the research to design and test the virus. That doesn't include the trial itself. Is probably close to $500,000 just to actually make that virus. And it's partly that expense is because you're going to give this to a patient. And so you have to be absolutely sure that that is safe, that it's not going to have any toxins in it, it's not going to have any infection in it, So these are some of the reasons why things are so expensive. But there's no question that funding is an issue, and probably will always be.
Hannah?
Yes, I believe you mentioned that the importance of detecting cancer at an early stage. You also mentioned that there's some genetic information that would indicate the probability of developing cancer. Would it make sense to establish a system whereby presumably the newborns are identified in this sense, to identify what kind of cancer they might be likely to develop, so that it would be then possible to establish a close monitoring in that particular area?
I couldn't totally hear the question.
Oh, okay. So we had a little bit of trouble hearing you at the front here. So can I confirm? So early diagnosis, and then trying to determine the probability of developing certain types of cancer?
Not exactly. What I'm saying is, I believe you mentioned that there's a genetic information that identifies the likelihood or the higher probability that an individual might develop certain type of cancer. And the question is, would it then make sense at some point to establish a system whereby you identify for presumably every newborn eventually, what are these genetic information, what is genetic information indicates, what sort of cancer that the individual might then may be facing, so that it would be possible then to concert a closer monitoring in that particular area.
Mm-hmm. So the challenge is that most of cancers do not develop because of an inherited mutation. For ovarian and breast, there's BRCA1 and BRCA2 mutations, and they increase risk. But if you put the whole population of ovarian cancer patients together, it only makes up about 15 to 20% at the very most of the population of ovarian cancer patients have that mutation. The rest are all what we call sporadic. There's no family history, there's no mutation that we could have looked for that would say that this woman is at higher risk for breast or ovarian cancer. And that's probably true. I don't know all of them, but there's very few other cancers that deviate from that. We don't have those mutations at birth that can predict reliably that you will get cancer.
I understand. And then is it then fair to say that, I believe you're saying that a majority of cancers are due to some random deviation from the normal for a given cell.
Yep.
Is it correct to say then that everybody has cancerous cells in their body?
Yeah.
But that normally, under normal condition, the immune system or some other system takes care of it. So therefore, the issue then is, would this is somewhat similar to a situation where if you have a bacterial infection, the body will take of it, but if the infection appears to be very intensive, then you develop sepsis. Would that be something similar?
It's actually very, well, it's actually very similar. So you're exposed to bacteria and viruses all the time, and without you even knowing it, your immune system fights off most of them. Occasionally, they take hold, and then you have an infection or of some sort, but cancer is exactly the same way. It's understood that if you were to look very, very, very carefully in every single person, if you could somehow do that, you could find cancer cells in everyone, but our immune systems are usually there to get rid of them very efficiently. It doesn't work as well as people age, right? That's part of the problem, and that's why cancer is a disease of aging, is because as we age, our immune systems don't function as well as they do when we're young. And so they don't fight off those cancer cells that might be appearing in our bodies.
Okay. We have about 5 minutes left for questions, so go ahead with yours, and then maybe one more. Oh, okay, there we go.
Thank you very much. What Dr. van der Heijden said about starting with only 3 ovarian cancer researchers really resonated with me, because I lost my mother to neuroendocrine tumor, carcinoid cancer, NET cancers. And even though NET cancers are apparently the fastest growing class of cancers worldwide, it really feels like very few researchers are involved in this type of cancer. So it really feels like, interest begets funding, and funding begets research. And I'm just wondering, from your perspective and your experience, do you have any advice for generating that interest which will lead to research for a cancer that is apparently fastly growing and yet very few people have even heard of it?
Yeah, including me. So you guys can address the cancer part, but the motivation actually is largely driven. The reason that I'm the Corinne Boyer Chair in Ovarian Cancer Research is because Patrick Boyer, who was married to Corinne Boyer, was very angry and frustrated when his wife died of ovarian cancer, and there was nothing in the country that he could find. He literally went from place to place across the country, found where we were, and started putting some money behind us in order to build the community. So it was done entirely by the husband of a woman who had died of this type of cancer. So that personal motivation is key. The other thing is there is a rare diseases group in Canada that is very active. It used to be, there's a strong component here at CHIO, and I think it's based in Vancouver. It's called the Rare Diseases Network. And so it tries to provide support and funding to do research on rare types of diseases, including rare cancers. So you might want to follow up with that. There might be some group there already, and maybe you know something more than I do. But
there's certainly, you're right in the sense that I do think that when cancers have a lot of patients affected, particularly if the outcomes are good, those patients become survivors and then they become advocates. It's sad when cancers are actually quite aggressive. Many of those patients die and they don't live long enough to advocate, but fortunately their family members will often. Neuroendocrine tumor does fall under the Rare Cancer Initiative, both in the US and Canada. But what I think is exciting is because we are becoming so much more global with connectedness, then there are a lot of opportunities for us to collaborate with one another across different countries. So I know several of our oncologists are very involved in ComNets, which is the Commonwealth group that addresses neuroendocrine tumors, and they do trials across all the Commonwealth countries and stuff because they aren't as common and not as easy to get patients into studies.
Yeah, I know that a few of our oncologists, like Rachel Goodwin, for example, they've been trying to establish databases of patients in that situation with neuroendocrine tumors. I know they've been flying around sharing that knowledge, and they are very interested in, what's next for that tumor type. And these have to be collaborative studies because no one site can actually, say, get enough patients to enroll. But that's where a academic group like the Canadian Cancer Trials Group is very important. So I'm one of the executive of the Canadian Cancer Trials Group. It's an academic group. It's the clinical trials arm of the Canadian Cancer Society, okay? And we meet in Toronto like twice a year, and we talk about, what trials will industry not fund, and where do we need funding to go to? What trial ideas do we have? This is how those kind of cancer types are addressed. So I think things are happening, but slower than you'd want. I treat, for example, uveal melanoma, a totally orphan disease site. Rebecca and I have talked about doing something together, but these are things that you try to do in your spare time because it's relatively rare. You see it enough. But yeah.
Okay, so one more question.
You're working with the immune system, and that's a good thing. Apparently, the immune system, people have stronger or weaker immune systems. If we strengthened our immune system, would that reduce the chance of having cancer or play some part in it?
Dr. Ong, is that one for you first?
Yeah, so we know that there's an association with suppressing the immune system and the development of cancer. And the most extreme example is when you have transplanted patients. So somebody with a donated kidney, for example, they get a kidney transplant, and we have to use immune suppressive medications that are very potent, that actually don't let T cells do their job, to prevent rejection, okay? And in those patients, there can be cancers like skin cancer that develop, or lymphomas. And what's impressive is that if we were to shut that immune suppression off, Sometimes the body just gets rid of those cancers once again. I have been very impressed with the immune responses amongst the elderly, amongst my patients. So it's actually not so much that patients that are older don't have immune systems. I think sometimes their immune systems just forgot. And you can reinvigorate them. And in fact, the responses can be stronger. So those are certainly thoughts that come into mind in the clinic.
Dr. Auer, I'm just going to get you to add a little bit as to immune system as it relates to surgery, which is something that you do a lot of work.
Yeah, so I mean, I think we do believe that when the immune system is suppressed, then cancers have an opportunity to grow and also to spread and to go to other places in the body and form tumors there. My area of research is focused on how surgery might, promote the spread of cancer. And sometimes when I say that, people get really worried, like you say, I shouldn't have my cancer surgery because the cancer could spread. And there's certainly that old wives' tale that if you let it to air, then it'll spread. And I think there's probably something to that. But cancer surgery is a very effective way of curing patients of cancer. I do think, though, around the time of surgery, we need to recognize the suppression of the immune system, and my research is really focused on stimulating the immune system before and after surgery to try and prevent that effect of immune suppression and the aftermath, including cancer spread. But I think that we're understanding more and more how the immune system is interacting with cancer cells, both before they become cancers, when we have the immune surveillance, which, again, most of us are, when we're protected from getting cancer, it's because our immune system has protected us. And what are the things that we can do lifestyle-wise or medication-wise that can promote a healthy immune system?
I'll steal the last question. Do we have the best cancer research and care team in Canada?
Yes.
Yes, okay. All right. I thought that was going to be the first question, but I made it the last. Thank you so much to everyone. Thank you again to our panelists. I think everybody agrees that what they've shared tonight, their stories, their advances, successes, and even challenges is very inspirational and informative. A very special thank you to the Taggart Parkes family and foundation for helping us make tonight possible for all of our guests and for patients for years to come, because we'll be sharing this information tonight, making it available for those who couldn't be with us. Alors, merci beaucoup encore une fois d'avoir été avec nous ce soir. I'd like to invite you to remain here for a few minutes. We still have some food and beverages for you. Take the time to mingle, kind of talk about the really awesome things that you heard tonight. Thanks again for joining us at the Ottawa Hospital, and have a wonderful evening.